All right, are there any questions? So last time I was talking about phytochrome, right, which is sensitive to red light and far red light, right? So we talked about this, like you have P red, right, red sensitive goes to, right, phytochrome far red sensitive, okay, and this was the active form, right? Right. Now the responses to phytochrome, they vary in lag time and escape time. So phytochrome responses vary in lag time and escape time. And I will explain what escape time is. Lag time is the amount of time that the phytochrome has to be activated before you see a response. Right? Escape time I will explain in a few minutes. Right? And examples of phytochrome responses are, for example, red light induced inhibition of stem elongation. Right? Remember, I told you last time that when the new seedling is growing underneath the ground, okay, it's in the dark, it's growing really, really fast in search of light. As soon as it sees light, the growth slows down, okay, and that is brought about by phytochrome. So red light induced inhibition of stem elongation, that particular response is observed within minutes. Right. On the other hand, phytochrome can also bring about induction of flowering, and that usually takes several weeks. Right. So induction of flowering occurs several weeks after red light exposure. Now, I have also talked about escape time. Okay, what is that? Well, if you apply red light, right, and then you're seeing some response, right, there is a certain amount of time up to which if you switch from red light to far red light, the response is reversed. Okay, and in the case of germination, you see that. Okay, but there is only a certain amount of time. If you wait too long, then you cannot reverse it. And then you say that it has escaped from reversibility. Okay. So the time that you can afford to wait before the response can be reversed, that is called the escape time. Right? So escape from photoreversibility so the red light effect can no longer be reversed by increasing the far red to red light ratio. Right? The ratio of far red light to red light is what determines whether you're going to have phytochrome in the PFR form, right, or in the PR form. Okay. Now, phytochrome, after the absorption of red light, right, it regulates many responses, right? So phytochrome regulates after absorption of red light. That means it has been converted into which form? After it is absorbed red light, right? Phytochrome is converted into which form? PR or PFR? PFR, right? So that's the active form, right? And how does it go about doing its activity? Well, it can do it in a couple of different ways. The first one is it can change membrane potentials and ion fluxes. And this response is typically going to be fast, right? because it's changing the membrane potential or the ion flux. It's not having to synthesize anything from scratch, right? So this is going to be a fast response. And the other response is that it can bring about a change in gene expression, right? So that has to go through the transcriptional machinery. It'll be a lot slower, right? So it can bring about changes in gene expression. So here, the response will be slower. And even when you're looking at changes of gene expression, right, some of the genes that are being transcribed, they themselves might be transcription factors. So you can have a primary response, right, and a secondary gene expression, right? So you could have a primary response. So if you're looking at gene expression, you can have primary response genes. And you can also have secondary response genes. Right? So maybe some of the primary response genes might serve as transcriptional factors, right, for another secondary response gene transcription. Now phytochrome is going to act on some proteins that are called phytochrome interacting factors. Abbreviated as PIFs. These guys act primarily as negative regulators of photomorphogenic responses, right? So these phytochrome interacting factors, they're acting like brakes on those photomorphogenic responses. That means responses or changes in form that take place in response to light, right? So when phytochrome in the activated form comes and binds it, it releases the brake and you get that photomorphogenic response, right? And I give you a few examples, right, of these kind of responses. The first one is seed germination that we already talked about. That is brought about by red light. That means the PFR form of phytochrome, right? Red germination. Another one is if light is there, then the plant needs to start photosynthesis, right? So it has to produce chlorophyll, right? Chlorophyll biosynthesis has to take place. So chlorophyll biosynthesis. Then shade avoidance. Then hypocotyl. Okay, so chlorophyll biosynthesis I already explained. What about shade avoidance? Plants like to avoid shade, right? So if you put them in shade, they'll try to grow towards the light, right? So that's also mediated by phytochrome and these phytochrome interacting factors. Then hypocotyl. Hypocotyl elongation. So if you look at a plant seed, right, typically if it's like a seed of lentil or something like that, there are two things that are called cotyledons, all right? That's the portion that we eat, right? And so when the seedling comes out, the portion below the cotyledons is called the hypocotyl. Hypo means below the cotyledon. That's the portion that really elongates real quickly in the dark, right? So if you give light, that's going to get arrested. Okay, that's another response that is brought about by phytochrome, right? Now, phytochrome signaling involves a number of different phenomena, right? For example, one is phosphorylation, dephosphorylation. We know that kinases, phosphatases, they can switch things on and off, right? So phytochrome signaling involves protein phosphorylation and dephosphorylation. and phytochrome induced photomorphogenesis involves protein degradation. Because remember, as I said, these pathways are negatively regulated. It's basically some brake that you're removing, right, to activate the procedure. So protein degradation, right, so these are negatively regulated. So there could be inactivation of a protein, degradation, or removal of repressor proteins. Right. So we finished our discussion of phytochromes, right? So they are the ones that basically respond to red and far red light, right? So now we will talk about other photoreceptors that respond to blue light. Okay. So blue light responses and photoreceptors. Right. So there are mainly three types of photoreceptors that respond mainly to blue light. Right. The first one of them is called cryptochromes. Then phototropins, and then there is another family that is the ZTL family of proteins. Right. So we will talk only about these two, like the first two over here, right? Not the last one. Okay. Now, have we already seen any blue light responses so far? Yeah, which one? Huh? Yeah, go ahead. Yeah, stomatal closure, right? Or stomatic opening in response to blue light. Okay, and you know the mechanics by which that is accomplished, right? So we already covered that. So that is actually brought about by phototropins, right? So let's talk about some of these things. So first, let's look at some of the general characteristics of blue light response. So one characteristic is that, see, for red light responses, we saw that the lag time was variable. Okay, it can be very quick, or it could be pretty slow, right? Like flowering versus, germination. Okay, one of them is fast, the other one is slow. You see the effect much later. Okay, so in the case of blue light, the responses, the lag time is a lot more, right? So significant lag time. And the response persists after stimulus is removed. And among the cryptochromes, right, there are two cryptochromes that are called Cry1 and Cry2, and these have different developmental effects. So Cry1 and Cry2 have different developmental effects. And let's see, for Cry1, right, this one actually controls a number of things. The first one is inhibition of hypocotyl elongation. That's what I talked about. It's controlled both by by red and by blue light. Okay, so hypocotyl elongation. Then it also controls the setting of what is called the circadian clock. See, just like we have a biological clock that allows us to keep track of time, right, night and day, even the plants do that, right? And the gene that plays an important role is the cryptochrome 1 gene, right? So this is a setting of the circadian clock. So basically training the clock to keep proper track of time, right? So setting up the circadian clock. And then the Cry2 gene, right? It plays an important role in the induction of flowering. And there are details associated with all these different pathways. Okay, they are spelled out in the book. And if you're focusing on that particular problem, you can study it. So that's our discussion of cryptochromes. Then the next group of blue light photoreceptors are the phototropins. So phototropins regulate chloroplast movement. Right? We have already seen that, if you have too much of light, the chloroplast will move to the side of the cell, right? And not absorb too much of light, which could be disastrous, right? So that is achieved by phototropins. So phototropins regulate chloroplast movement. In addition to that, they are the ones that are responsible for blue light mediated stomatal opening and blue light mediated stomatal opening. And by now we have some idea about how that occurs, right? So phototropin irradiated with blue light starts a signal transduction cascade, which ultimately results in the activation of H plus ATPase. Right, and this we have already talked about, right? And how the pH increases, then potassium flows in and so on, right? Okay, that's the discussion of phototropins. And then the next thing is, about the next one, we don't have much of a discussion, because it's a one that has been just recently found. So the photoreceptor responsible for UVB induced, so ultraviolet light B, okay, it can be of three different types, A, B, C. C is mostly absorbed by the ozone layer, right? A and B reach the surface of the earth, right? So the photoreceptor responsible for UVB induced developmental processes is called UVR8, right? And this has been recently discovered. And unlike the others, it doesn't have a chromophore. See, like the other photoreceptors we are talking about, it has got a protein and it has got a chromophore. Okay, this one is just a protein. So recently discovered and lacks a chromophore. Has only a protein. All right, any questions? So otherwise we'll now move on to the next chapter, which is chapter 17. And this is embryogenesis. So embryogenesis is the process of producing a seed from a single fertilized egg. So single fertilized cell, which is called a zygote. So again, I mean, during embryogenesis also it's control of gene expression, right? Regulatory networks at play, right? That will finally produce the seed. So let's make a number of observations. Regulated cell divisions produce the polar axis, right? Because you have to have an axis, okay? One way you're going to have the shoot, the other way the root, and so on, okay? So the cells have to divide in a particular pattern, right? So regulated cell divisions produce the polar axis, and bilateral symmetry of the embryo. The second thing to note is that plant growth is indeterminate, right? If you are, let's say, a human being, right, and you're born like I mean, before birth also, right, when you develop from a single fertilized egg, you'll have the right number of fingers, right, right number of ears, and so on, okay? In the case of plants, depending on the environment, it might shoot out another branch here, there, okay? So it's indeterminate, right? It has to have because it's a sessile organism, it's in one place, right? So it has to be able to adapt to the environment, right? Okay? So it's different that way. So the second point to note is that plant growth is indeterminate. So there has to be flexibility to produce new branches, leaves, etc., depending on the environment, right? So plants are sessile organisms, and therefore have to maintain the capability to grow adaptively to respond to the environment. So, and how do plants do that? They'll basically keep what are called meristems, okay, where they store some stem cells, okay. So as needed, they'll draw cells from there, right, and produce new structures, right. So post embryonic plants retain meristems or stem cell niches. Now these meristematic cells, so meristematic cells are undetermined and are central to plant growth and development. Now in the case of plant growth, all right, there are three major stages of development. So three major stages of plant development. The first one is embryogenesis. Or seed formation. The second one is what is called vegetative development. And this includes germination. So the seed has to germinate. Then there has to be a seedling. So seedling establishment. Organ formation. Etc. And then the last one is reproductive development. So in this chapter we are focusing only on embryogenesis. Okay, that is the formation of the seed, right? Starting from the fertilized cell or the zygote. So now let's talk a little bit about the origins of the polarity in the seedling, right? I mean, how is polarity established in the seed, right? So the origins of polarity, and obviously signaling is going to determine, okay, which side is going to have the cells for that will lead to the shoot versus the root and so on, okay? So the apical, so point number one, the apical basal polarity. Apical is towards the apex, right? So you'll have a shoot apex at the top, root apex at the bottom, right? And then you have the base, right? So the apical basal polarity is established early in embryogenesis. And here you have auxin is an important signaling molecule, right? So auxin functions as a mobile chemical signal during embryogenesis. So if you go ahead and tinker with the auxin flow or auxin concentration, you're going to be able to move structures around. Okay, things are going to change. Then also intercellular signaling processes play key roles in guiding position dependent development. And even the cell division, right, it takes place in two different directions to produce these different structures, right? So for example, if you have, let's say, a layer of cells, like along the annulus over here, right? So you have these cells, right? Then when these cells divide, right, there are two options about the direction in which the cell wall is going to be placed, right? So if the cell wall is placed like this, right, in that direction, that is referred to as periclinal division, right? Periclinal division. So you are literally adding an extra layer, right? So if each cell divides in a periclinal fashion, you basically now have one more layer of cells, right? So periclinal division. On the other hand, you could have a situation where the cell wall actually divides, right, perpendicular to the direction of, let's say, the circumference, right? And that's called anticlinal division. Like you could have a situation like this here. So that would be an anticlinal division. And there are signals that will determine which kind of division is going to take place, because you have to build up a structure, right? Starting from like a few cells, and you have to grow on that. Okay, so things have to happen in the right way, right? But these are two possibilities for cell division, right? Now typically, right, how do you get I mean, how do you finally end up with the seed starting from the fertilized egg or the zygote, right? So I'm just trying to give you a brief sketch. There are more details in the book, all right, all precise details. Okay, so what I discuss here is not going to be very precise, but it'll give you the big idea, right, or the big picture. So you have a single cell, right? This is called the zygote. It has been fertilized, right? So the male and female gametes have combined, right? And on one side you have a large vacuole. So this cell then splits in two, right? On one side there is a small cell which is called an apical cell. The other side there is a large cell, right? That's called the basal daughter cell. So it splits in two, right? So you have the apical cell here, which is small. So apical daughter cell. And then you have the large basal cell. So large basal daughter cell. Then in the next step, this apical cell is going to split into 4, then to 8, and so on, then into 16, right? And most of the stuff actually comes from there, right? So in the next step, you get 8 cells here, right? So the basal cell doesn't do anything much, but this guy you're getting like maybe 8 cells. And again, I mean, the orientation has to be proper, right? And I cannot draw a good picture here. You can look in the book, all right? So you'll get 8 cells, right? From the apical cell, right, apical daughter cell. Then after that, in the next step, right, there will be 2 layers of actually 16 cells in the apical region. So here, from that single apical daughter cell, now you have 16 cells, and these are arranged in 2 layers, all right, like this, something like this, all right, and then more cells here. There are 16 cells, right? Now, the top layer over here, right, of these apical cells, that will give rise to the shoot apical meristem. That means the bunch of stem cells, the niche of stem cells that will give rise to the shoot. This is all happening in the seed. So this is shoot apical meristem, right? Then also the cotyledons. Like if you look at the beans, okay, you split them into those things that are called cotyledons, right, in the beans, right? So it will give rise to that. Then the second tier over here is going to give rise to the stuff underneath the cotyledon. Okay, so that's the hypocotyl, all right? Hypo means below, so hypocotyl will give rise to that. Then it will also give rise to the root, right? And the upper cells of the root apical meristem. Again, the bunch of stem cells in the root, all right? So root apical. Meristem. And then these guys here, right, they give rise to some of the other structures, right? So this becomes what is called a suspensor, right? And it provides the uppermost cell of this suspensor, right? So this also undergoes a bunch of cell divisions. So the uppermost cell becomes what is called the hypophysis. And this hypophysis divides to form what is called the quiescent center. And I will show these in a picture of the root a little bit later. Quiescent center and the stem cells. And the stem cells, which are called initials, that form the root cap. Because you do see the tip of the root, all right, it needs to grow into the soil, all right, so you need a bunch of stem cells over there, okay, and that collection over there is called the root cap, right? So, and I will show, I'll draw a picture showing all these different regions in the root, right? Now again, I mean, as engineers, for us the take-home message is that all of this is driven by signaling, right? So there is communication. So embryo development features regulated communication between cells. And by analyzing mutants, like you can go and mutate some of the genes, okay, you can see abnormal embryos that will develop, right? And then you can figure out which gene plays which role, right? For example, something that is supposed to develop into the shoot apical meristem, if you go and mess up those genes, you won't have any shoot apical meristem, right? The shoot will be missing. Root could be missing, right? And both could be missing, right? You can try all kinds of things, and people have already done those, right? So the analysis of mutants identifies genes for signaling processes that are essential for embryo organization. Example, missing shoot apical meristem root apical meristem or both. And plant polarity, again auxin is at the center. Okay, there are auxin streams that will determine where the growth is going to take place, right? So plant polarity is maintained by polar auxin streams. And here, when you're talking about polar auxin streams, the auxin basically moves by crossing the plasma membrane. So it's not symplastic transport or apoplastic transport, right? It is actually crossing the plasma membrane. So that is transport is carried out. So there will have to be auxin transporters, right? Transporter proteins that are involved. So that is transport is carried out by crossing the neighboring cell membranes. Now auxin uptake by cells, that means the auxin entering cells, and the efflux from cells, auxin leaving cells. So and efflux from cells are regulated via multiple mechanisms. And involve various genes. And this can be studied by using GFP reporters of auxin transport proteins. Remember I talked to you about green fluorescent protein promoter reporter technology, right? Basically you can follow the activity of genes. So this can be done, right? And people have, plant biologists have already done that, right? You want to see how auxin is moving, you can put in some auxin transport protein, put a tag on it, using a green fluorescent reporter, then you can see how it's moving. Right. All right, a few more observations. And this is something that I said before, but I want to re-emphasize. The meristematic tissues provide the foundation for indeterminate growth. And as I have been emphasizing, this is very important for plants because they cannot move around from one place to the other, right? So in the root and the shoot meristems, there is a cluster of slowly dividing and undetermined cells called initials, and these are the ones, like, they are basically stem cells. Okay, they keep the ability to differentiate into any type of cell, right? So according to the needs of the moment, they are going to differentiate depending on the environmental conditions, right? All right, any questions? So then I move on to the next chapter, which is chapter 18. Yeah, we have 24 chapters, so we are getting there, okay, slowly, right? So chapter 18, so seed dormancy, germination, and seedling establishment. So what we discussed in the last chapter, that is what is needed for producing the seed with all those meristems and things like that in it, right? Now once the seed has been produced, then it's kind of dried up, it's being desiccated, right? It can be transported over long distances and it enters a period of dormancy, right? So normally, the seed is going to enter a period of dormancy, so it's not going to germinate right away. Now, if there are seeds that don't enter a period of dormancy, then you may have a situation where on the parent plant, okay, you can see the seed is again producing a seedling, right? That can happen too, right? But in most cases, that does not happen. So once the seed has been dried, it has been transported over long distances, right? And then, in order to germinate, it needs water, right? Water, light, and other conditions, right? So seeds require hydration and sometimes additional treatments. And I'll tell you what they are. To germinate. Now during germination, see, once you have a seed, you're going to have a plant grow out from it, okay? Until that seedling, okay, develops green leaves, it is not able to carry out any photosynthesis, right? So whatever nutrition it has to get from the stored nutrition in the seed itself, right? During the period prior to it becoming what is called autotrophic, that means capable of producing its own food, it has to get the nutrition from the seed, right? So during germination and seedling establishment, food reserves sustain the plant until it is autotrophic. That means capable of producing its own food. Now the seed dormancy may arise from the embryo itself. So the embryo itself may have gone to sleep, okay? It will wake up after some time, in response to the right stimulus, or from the surrounding tissues. Such as the endosperm. So the endosperm is basically it has got starch and other things in it, that's the nutritional tissue in the seed, right? So such as the endosperm and seed coat. Now seeds that don't become dormant, as I said, they could germinate on the original plant itself, right? So seeds that do not become dormant can exhibit what is called vivipary and precocious germination. That means seed germinating on the original parent itself or original parent plant itself. Now how does that germination happen? Again, there is signaling involved, right? Abscisic acid and gibberellins, okay, these two interact. So the ratio of these two hormones determines whether it's going to germinate or not, right? So, I mean, there's a reason why I'm covering all the details of the biology, okay, because so that we can get to the signaling part, right? So abscisic acid, which is a plant hormone, so abscisic acid actually promotes seed dormancy. Right? While gibberellins break seed dormancy. And there is a reason, seeds should have some dormancy because suppose, in the fall, towards the end of fall, right, the seeds have been produced, they germinate right away, right? What's going to happen? The winter frost is going to come and kill everything, right? So there are seeds that need to be exposed to a period of cold before they start germinating, before you can germinate them successfully, right? So they are keeping track of seasons too, right? So gibberellins break seed dormancy. And how do they do that? Well, this abscisic acid or ABA, right, abbreviated ABA, it inhibits alpha amylase transcription, right? So what do you think the amylase is going to do? It is going to break, amylase means it's going to break down amylose, right? Some kind of sugar, right? Remember, the seed needs to eat, right? During before it becomes autotrophic, it needs to eat, right? So it has to be able to break down starch to produce sugars that it needs, right? So this ABA, abscisic acid, it inhibits the transcription of the enzyme that is going to be needed to break down starch, right? So ABA inhibits alpha amylase transcription while gibberellins, this hormone called gibberellin, so gibberellins enhance alpha amylase transcription. And as I said, alpha amylase protein breaks down starch in the endosperm and the resulting sugars can be used for nutritional purposes by the seedling, right, by the seedling that is developing. Can be used for nutritional Now, how is the release from seed dormancy brought about, right? So there are a number of stimuli that can bring that about. So release from seed dormancy. One of them is light. Okay, the presence of light, it can bring about an end to seed dormancy. Another one is chilling or after ripening. So some seeds, unless you kind of chill them, right, or get them close to freezing, expose them to cold temperatures for a certain period of time, you try germinating them, it's not going to work, right? So chilling or after ripening, and so, or you might have to expose them to room temperature for some time, after the seed has ripened, expose it to room temperature, and then only it's going to have the capability to germinate, right? And this allows it to, the seed to, if you're talking about chilling, it can keep track of seasons, right? So can track seasons. And another stimulus that can be used to to break the seed dormancy, another set of stimuli are nitrates, so nitrate, nitric oxide, and smoke can break dormancy. I wonder whether the smoke has anything to do with, plants uh, germinating and all that, or new plants being produced after a forest fire or something. I don't know. I'm just making a conjecture. Since smoke is listed here as one of the items. Right? Now, germination takes place in 3 stages. Or in 3 phases relating to water uptake. So the first phase is when the plant is, the seed is going to absorb a lot of water, right? It swell up, then it will break the seed coat, and then something comes out, right? That's the first phase. A lot of water is absorbed. In the second phase, less water is absorbed because the seed now has become metabolically active, right? And in the third phase, again, there is a drop of water potential, and more water is drawn by the seed, right? So phase 1, phase 2, and phase 3. Phase 2 and phase 3. So this is less water absorbed in phase 2. All right, next I want to talk about tropisms, or the ability, right, of a growing plant to go in the direction of light, right? Go in the direction of gravity and so on. The roots need to go downward, the shoots need to go upward, right? So different stimuli is going to direct the development of the plant. So tropisms, so what are tropisms? Tropisms are growth in response to directional stimuli. And again, auxin plays a very important role, right? So at optimum concentrations, Auxin is a growth factor for the shoots, all right, and it is a growth suppressor for the roots at optimum concentrations, all right. At higher concentrations, things can change. So at optimum concentrations, auxin promotes stem and, let's see, coleoptile, right? Coleoptile. Okay, and what is that? That is the first leaf. Coleoptile is the first leaf above ground in grasses. So it's something like a stem, okay? So first leaf above ground in grasses, right? When grasses start growing. So that's called coleoptile. So in stem and coleoptile, auxin is going to promote growth and it inhibits root growth. Right? So let's look at a couple of scenarios, okay, to see how the tropism is going to occur, right? So if you have a coleoptile, right, and let's say the auxin, right, goes to the bottom, right? So if auxin goes to the bottom, so what is going to happen? It's a laterally placed coleoptile or a horizontally placed one. Auxin has gone to the bottom, right? So is this thing going to bend? More auxin at the bottom, right? So which way is it going to bend? So somehow there is more auxin at the bottom than at the top. So the growth will be more at the bottom or at the top? Bottom, because this is a coleoptile or a stem, right? So that means this is going to bend upward. That's what you want in the shoot or in the grass, yeah. So it will bend upward. On the other hand, if you have a root, right? So if you have a root, then same thing happened, right? So this is root, all right? And let's say the auxin went to the bottom. So what is going to happen here? If auxin goes to the bottom, right? More auxin at the bottom, right, than at the top. So then the bottom will grow less or more? Hmm? It will go down because the bottom is going to grow less. Okay, in the case of roots, it's going to inhibit the growth, right? So it's going to bend downwards, right? So this is, you're seeing gravitropism, right? Right, it's going to move towards gravity, right, in the direction of gravity, right? Then there are additional things also that direct, root movement downwards, all right? There are things called, like, see, for example, if you want to see the direction of gravity, all right, if you take a solution, all right, and you have some molecules that are put in there, right, they will sediment towards the bottom, right? So if you didn't know, which direction gravity was, if you had a solution and you saw things sedimenting, you would know the direction of gravity, right? Plants use that, right? So in fact, there are things that are called statolites, right? Statolites. Uh, and let me just finish writing the sentence, then I will explain the details, okay? So statolites in special cells that are called statocytes. Statocytes serve as gravity sensors in root caps. So the root cap is something that is at the tip of the root, and that's the one that decides, in which direction the root is going to go, right? So what are these statolites and statocytes, okay? Well, statocytes are specialized gravity sensing cells, right? And statolites, these are what are called, these are these are amyloplasts that function as gravity sensors. So amyloplasts that function as gravity sensors. And what are amyloplasts? These are basically organelles or plastids that contain amylose or starch. Okay, I mean amylose is a component of starch. So amyloplasts, these are starch containing plastids. So again, it gets back to the same idea that I mentioned. If you have a solution, you have molecules, they go to the bottom, that's the direction of gravity. That's how the root sense gravity, right? Any questions? So next I want to talk about phototropism, right? I have been referring to auxin, okay? How does auxin make sure that the plant is going to grow towards light, right? So phototropism. So phototropism involves internal redistribution of auxin. All right, so if you have a situation like this, all right, so first let's assume that a plant is being exposed to light on both sides, right? So then you have a situation like this where you have the cotyledons and then there is the stuff downward. See, the auxin is produced at the tip of the shoot, right? So it moves downward from the top to bottom, okay? So let's say these are the cotyledons that you have. So these are the two cotyledons, all right? And then auxin is going to flow downwards, right? So it's going to flow downwards like this. It goes from the tip of the plant downwards, and even the root gets its auxin coming from the shoot, right? So this is the normal auxin flow in the dark, right? So in the dark, the auxin flow is from shoot to root through vascular tissues. And what kind of vascular tissues are these going to be? What kind of vascular tissues do we have in plants? Phloem, yeah. So this has to be phloem, yeah, phloem tissue, right? Okay, so now what you do is you expose one side, right? The left side, let's say, to blue light, right? Right? Left side of the plant is exposed to blue light, the right side is still kept in the dark, right? Then what happens is that initially that flow, the downward flow, it actually stops at the cotyledons, right? So it is initially it's going to just stop here. You're not going to have a downward flow, so it just stops here, right? And then more of the auxin is going to get distributed to the dark side, right? So flow stops here, and then flow shifts to the right side. So still you're giving the left side blue light. So now there's a little bit over here and there's tons of auxin on the right. So flow shifts to shaded side. Now auxin is a growth hormone, right? If the flow shifts to the right side, that means the right side is going to grow more, right? So now the plant is going to bend in the direction of light. So there you're going to have bending now. So again, still exposed to blue light, and the plant bends like this. And now you're having like a downward flow on the left, and then a much thicker flow on the right, right? So that way the growth on the right side is going to be much more. So the plant bends. All right, so this is how auxin brings about phototropism, right, in plants. Any questions? Now I want to talk about what is called photomorphogenesis, right? so which basically means change in form, right, in response to light, right? Now seedlings transition from scotomorphogenesis, right? Scotomorphogenesis is growing in the dark, right? So scotomorphogenesis, which is basically development in the dark. Which usually means underground. So that is underground. There's no light over there. To photomorphogenesis, so development in the presence of light. At the first instance of light, if you just give it a flash of light, that's going to happen. One flash, you don't have to, doesn't have to be sustained, right? So at the first instance of light. So light is like the stimulus, right? You have all these signaling pathways, light just stimulates that and particular pathway is activated, right? And what are the hormones that are involved here? There are a number of hormones, gibberellins and brasinosteroids. They suppress photomorphogenesis in etiolated shoots. Etiolated shoots means basically before it's been exposed to the light, right? So etiolated shoots. And you've all probably seen, germination take place, how a seedling grows. Initially it's bent, then the leaves are also closed and all that stuff. And that is etiolated. So, and it'll not have the green color also because it's not exposed to light, okay? So these two guys, gibberellins and brasinosteroids, they are going to suppress photomorphogenesis. That means converting to the form that you get in the presence of light, right? Now phytochrome, which is one of the photoreceptors, then auxin and ethylene regulate hook opening and lateral cell expansion. So usually the seedling will be in the form of a hook, okay, with the two leaves together, right? And this needs to open out. And the reason it's in the form of a hook is because one side is growing more than the other. So in the presence of light, this is equalized, and then they start growing at the same rate, right? Or the side that was growing slowly that catches up, right? And or actually the side that was growing too quickly that slows down, and then it becomes straight, right? So phytochrome, auxin, and ethylene regulate hook opening. And lateral cell expansion. So next I want to talk about shade avoidance. So photosynthetic plants, they will usually try to avoid shade. Okay, if there's a lot of shade, it'll try to grow taller, right? So that it can access the sun, right? So phytochrome actually mediates this response. So phytochrome mediates growth towards optimal light conditions in sun plants. And this is going to happen. Sun plants means plants that grow really well in the presence of sunlight. Okay. So if you have a low PFR, so in the shade you'll have a low PFR to PR ratio, right? Because you need red light to convert the phytochrome to the PFR form, right? That's what you want, right? So if you have a low PFR to PR ratio, That means the plant, in that case, the plant should try to grow towards light. It doesn't have enough of red light, right? So grow towards light. And you can actually modulate that. See, like if a plant likes to avoid shade, okay, and you're growing a lot of plants together, okay, they're shading each other, then the plant is going to expend most of its energy trying to grow taller than the other plants to reach the sunlight, right? So if you can go ahead and tinker with the genes, right, and make it such that the plant doesn't mind growing in some shade, right? Or you slow down its shade avoidance response, you can dramatically increase the yield because the plant is not going to grow, keep on growing just in search of more light. Okay, it'll do with whatever light that it has, right? And people have done that. They have increased the yield of maize by tinkering with the shade avoidance response, right? So increased tolerance to crowding. Yeah, yeah. So increased tolerance to crowding, that is reduced. Because at the end of the day, as far as the plants are concerned, what you care is about how much corn it produces, right? Right. So you don't care how much light it's getting, okay? So if it's just chasing light, then it's just going to keep going taller, right? So, mhm, yeah. So that is reduced shade avoidance responses has led to increased crop yields in maize plants. All right, another thing that I want to point out is the role of auxin and cytokinin for root difference, for development of root hairs and so on. Okay, I mean there are many different aspects. I'm just speaking on some of these, all right, to show you that all this signaling is extremely, extremely important. All right, so auxin and cytokinin are required for normal vascular development. And root growth and differentiation also is affected by hormones. And I have been promising you all along that I'll draw a detailed structure of a root, right? I'm going to do that right now to just show you where the root cap is. So typical root will look like this. So you're going to have different zones, all right? So this is the root. At the bottom of the root, you have something that is called the root cap, right? Then you have a small area where the cells don't divide, right? This is called the quiescent center. Then the zone, this quiescent center is present inside what is called the meristematic zone. These are the stem cells, right? So meristematic, because that's where you expect the growth, right? I mean, if it's digging deeper and deeper into the ground. So this is the meristematic zone. Then above that you have what is called the elongation zone. And still above that you have the differentiated zone. Right? And typically, in the differentiated zone, right, and the elongation zone, that's where you'll get things like root hairs, like lateral growths coming out, right, from here. So these are the root hairs. And root hairs, they play an important role because they can increase the surface area for absorption of water and nutrients and so on. Okay. So auxin and other hormones, and again, this stuff also is controlled by different hormones, right? So auxin and other hormones regulate root hair development, and in fact the places where the root hair develops, those have very high levels of auxin, right? So auxin maxima correspond to lateral region, so to regions of lateral root emergence. All right, now that finishes chapter 18. Right, I'll just start with chapter 19 and then I'll stop. So chapter 19, discuss a couple of things. So chapter 19 is about, so we have talked about seedling establishment, right? Then seedling establishment, then we talked about germination. So this chapter is about vegetative growth and organogenesis. Right? So again, the big picture, right? The take-home message is that is also controlled by gene expression and the activity of different hormones, all right? So controlled by molecular interactions and regulatory feedback. So different genes and transcription factors are involved. For example, if you're looking at leaves, right? If you're looking at leaves, there'll be an upper side of the leaf and a lower side of the leaf, right? So this is called the adaxial side. That means the upper side of the leaf, right? The plain leaves that you see on plants. So adaxial side, and at the bottom of the leaf you have the abaxial side, right? So there will be genes, right, and flow of hormones and all that that will determine, like, so examples of things that are controlled in this way would be adaxial, abaxial polarity, right? Which are the cells that are going to go on the upper side versus the lower side, right? Then where are the stomata going to be placed, okay? What is the stomatal patterning going to look like? Because there have to be pores, there have to be guard cells and so on, right? So stomatal patterning. So lots of things have to interact, right, in order to make sure that the stomata and all are formed in the right way, right? Then the pattern of the vasculature in the leaves, right, the phloem in the leaves, right? So it's called the venation pattern. So venation venation patterns. Sorry, it's venation. So venation patterns in leaves. Again, this is regulated by auxin canalization. And I want to just end by talking about shoot branching and architecture. Actually, this chapter is easy to finish. I'll just mention with one thing, right? And architecture. I can even go over this next time if necessary. See, the thing is, if a plant is growing, okay, should it keep on going up or should it branch, okay? That's the decision the plant has to make, all right? So if it keeps on growing in the direction of the apex, that is called apical dominance, all right? And if it starts branching on the sides, that's called lateral dominance. So that is brought about by these hormones called auxin and so on. Okay, so if you go and cut the top of the plant off, right, the auxin flow stops and then you have branching. So I will go over this at the beginning of the next class. That's the only thing that I had left to cover in this chapter. But I can quickly cover that in 5 minutes next time. So let's stop here for today.